Good morning, good afternoon, good evening, wherever you are joining us from, and a very warm welcome to our mini seminar. During this session, we are going to hear from exciting startups and emerging technology coming out from Japan. Before that, we would like to introduce ourselves. I'm going to hand over to Akihiko Soyama, President and CEO Director from Rickjay. Soyama-san, please.
Thank you for the introduction. I'm Aki Soyama of Rickjay. In next 2 minutes, I'd like to share the overview of Rickjay. Rickjay's official name is Life Science Innovation Network Japan. L means in a wide range of life science. I means to support and promote innovation. N and K by creating a network or ecosystem. J both in Japan and abroad. So Rickjay was established about 5.5 years ago, and we have now about 490 members. So to support and promote innovation, we have hosted a lot of events and programs such as big symposium, seminars, as well as acceleration program, incubation programs, and pitching events with our members. As for the network for innovation, we have collaborated with many global institutions listed in this slide. And we are expanding this network furthermore. So also we have collaborated with many Japanese institutions such as major universities, national research institutions, as well as many innovation hub cities and organizations. I hope you will enjoy this mini seminar and expand your and strengthen your network with Japanese startups and life science community. Thank you.
Thank you. Now we're proceeding to the main session today with having presentation from each delegate. I'm going to hand over to the first speaker, Mr. Takumitsu Akutsu, Director Corporate Planning from Quoripus. Akutsu-san, please start your presentation.
Hello everyone. So I want to start my presentation. This is Akutsu. I'm very excited to introduce my product. So iPSC-derived cardiomyocyte patch. So we believe the product will help the patient in heart failure, not only in Japan, but globally. We are Quoripus. So Quoripus comes from the cuore, heart in Italy, and the iPS. We combine these words as a name. Our goal are very simple and strong: save heart failure patient with regenerative medicine, cell therapy technology. This is a summary of our company. So our leading company using iPS-derived cell therapy for heart failure. We have first iPSC-derived cardiomyocyte product from Osaka University. Third, we already established stable and robust manufacturing process and facility for commercialization with Japan quality. And in the end, so we have a lot of partnership with diverse corporate for next pipeline and in the future growth. About us, we established March 2017. Our CEO Takayuki Kusanagi, MBA holder from Harvard Business School. He had a great background for the finance, investment, and business. We have 3 facilities: head office in Tokyo, and Osaka lab in Osaka University campus. And we established our own manufacturing facility near Osaka University. I want to emphasize our current topic. So we conducted a private placement over $30 million from last year. So we are really happy to announce that Professor Yoshiki Sawa is appointed as new CTO. We leverage this core technology from best academia in Japan and globally, the Osaka University. We have an exclusive right to use the technology and know-how from them. It's great. And Professor Yoshiki Sawa, he is both cardiovascular surgeon and a scientist. He led a long time as a head of the cardiovascular surgery department, Osaka University. And he also president of the Japan Society of Regenerative Medicine until this year. He had a lot of experience in research and insight about regenerative medicine and cell therapy field for over 20 years. We are really happy. He is a founder and the CTO of our company. Osaka University has a key iPSC technology accumulated from 2008. For example, the living cardiomyocyte from iPS cells, large-scale cell culture, purification and removing anaplastic cells, tumorigenic control, and a low-risk approach to arrhythmia. It's also important for the safety. He always emphasized bench to bedside. He also always asks us, what is truth? Truth is science. So we are really excited to work with him and his group. This is a quick profile for the iPSC-derived CM product. We culture iPS cells from the Kyoto University and differentiation to the cardiomyocytes. And then so we product a seed, iPS-CM patch, with a cell seed technology from Tokyo Women's Medical University. It is great news from the Osaka University. They conducted the first in-human clinical trial for this product from January 2020. This is a doctor-initiated clinical trial for ICM, and then so they already conducted the 3 patients in SAFE. They now evaluated the results of the clinical trial, and then so they will proceed the next step, more patient and a multi-center study. We are really looking forward to following up the result, and so they will report out very soon. So we are really excited about this. We established stable and robust protocol for safety and scale for commercialization. So like this kind of protocol, cell culture, differentiation, purification, freeze, and the production. So we have technologies from Osaka University, and so we can deliver a cardiomyocyte from iPS cells over 90%, and we can large scale cell culture over 1 billion, purification and removing anaplastic cells, and tumorologic control and arrhythmia control is really key for the safety. For the facility side, so we already established a new cell processing center called Quick One for the commercialization for our product. So we start to production, and so we are really looking forward to to deliver the patient. For the investor side, so as you can see, we have a lot of investors, not only the venture capital, but also corporate investors. Daiichi Sankyo, Mitsubishi Corporation, Thermo, Fujifilm, Asahi Intech, it's a catheter company, and the Taiko, Daidan Stem Cell Source. They already have a unique technology and the resources. Through the collaboration, we created a new pipeline and a new second and third product with them. We are really excited to collaborate with them. And also we are still seeking a new opportunity for the collaboration, not only in Japan and globally. If you have some interest, please contact us, and so we are looking forward to collaborate with you. Summarize again, so we are a leading company using state-of-the-art iPS-derived cell therapy for heart failure, and we have first iPSC-derived CM product from Osaka University. And so we already established a stable and robust manufacturing process and facility with Japan quality. And in the end, so we are really love partnership with diverse corporate for the next pipeline and the future growth. I really appreciate your attention. So we are really looking forward to working with you. Thanks.
Thank you very much for your presentation. I would now like to introduce the next speaker, Dr. Haruka Yoshie, Application Specialist from Cellink. Yoshie-san, please start your presentation.
Hello everyone, thank you for watching this video. My name is Haruka Yoshie. I am an application specialist at Cellink Group and the Biko company. Biko is a bioconversions company and offer a wide range of technologies, products, and services, and more than 2,000 labs worldwide are equipped with our technologies and products now. Cellink Group focuses on bioprinting related products and services. And in this session, I will focus on Cellink technologies and product. In this video, I will first do a brief introduction of 3D bioprinting technologies, followed by our extrusion-based printers and its applications. And then I will move on to the bioinks, the materials that are used for 3D bioprinting. Then I will spend the last few minutes of the presentation on our light-based bioprinters. There are several different 3D bioprinting technologies as shown here. The technologies that we offer are extrusion-based and light-based stereolithography technologies. With our technologies, you can have cells added to the materials, and with this cell-laden bioinks, you can print the live cells. For extrusion-based bioprinters, we use pneumatic systems so the air pressure pushes inks out of the nozzle or needles. These are the printers that we have. Here are some of the research work done using our extrusion bioprinters. The top left here is a work showing the cornea model printed with a fresh printing technique. Some researchers study infectious diseases using 3D bioprinting technologies, as shown in this publication. This one is influenza. 3D bioprinting technologies can also be applied to study the current pandemic, COVID-19. It has a broad range of applications, and these applications include organ-specific models, disease studies. Some of the researchers use 3D bioprinting technology to study cancer. For example, tumor microenvironment. It can also be used to fabricate microfluidic devices. Organ-on-a-chip is a rapidly expanding field. Drug testing and personalized medicine are another great applications. In the following few slides, I'd like to talk about bioink, as it is very important for the materials you're printing with to meet all the requirements for bioprinting. In case of extrusion-based printing, the bioink needs to be extruded out of nozzle in a controlled manner, and it also needs to maintain the shape once it is being extruded. Additionally, when bioprinting, where you work with the live cells, cytocompatibility needs to be considered. It is important for bioink to create an appropriate microenvironment for the cells. So what makes a bioink? It typically has a main polymer component, and other components can also be added depending on needs and interest. These include thickeners to improve printability and shape fidelity, bioactive components like growth factors to enhance bioink's interaction with the cells. Complementary reagents or chemicals like photoinitiators or neutralization buffer sometimes need to be added as well. The main polymers can be natural polymers or semi-synthetic polymers. These are some examples. And it is also very important to consider cross-linking methods when formulating bioinks, as cross-linking plays a key role in preservation of 3D construct shape after being printed. Cellink, having started as a bioink company, offers a wide range of bioinks. Please check our website to find out more about our bioinks. For the next few minutes, I'd like to briefly talk about our light-based bioprinters. Our Luminex uses DLP, digital light processing technology. And with this technology, you can print smaller and more complex structures. Shown here are some of the structures printed with Luminex. In this vessel exchanger model on the left, you have a hydrogel construct with two intertwined channel structures inside, and these two channels can exchange gas, fluids, and nutrients as they can travel through these hydrogels. Next is a vessel exchanger model. There is a vascularized alveolar model to study our lungs. If you'd like to achieve even higher printing resolution, we have Holographex, which uses multi-photon stereolithography technology. As you can see in these images, you can print very small structures. As a bioconvergence company, we have many other products that were not covered in this video. Please again visit our website for more information. We also have LinkedIn and Twitter. We look forward to connecting with you. Thank you for your attention. I wish you all a great day. Happy bioprinting!
Thank you very much for your presentation. I'm going to hand over to the next speaker, Dr. Shin Hato, CEO from Solution Inc. Hato-san, please start your presentation.
Hi there, I'm Shin Hato, the CEO of our company, Solution. Solution is a university-based startup company from Keio University School of Medicine. My background is ophthalmologist, and my specialty is corneal transplant. Today, I would like to introduce our project, Corneal Endothelial Regenerative Medicine with iPS cell-derived endothelial cells. There are unmet needs in corneal transplants. It is reported that more than 30 million patients are waiting for corneal transplants globally. However, only 180,000 corneal transplants per year are conducted. So the demand of corneal transplants are over 70 times the supply. And you can see the clouded cornea here. This is bullous keratopathy, corneal endothelial cell dysfunction, and progressive blindness disease. Bullous keratopathy is the most common disease for corneal transplants. More than 50% of corneal transplants are for this disease. Current treatment of bullous keratopathy is penetrating keratopathy or endothelial keratopathy. Although surgical procedures have been progressed, there still be many complications, and there still be many very difficult surgeries. So what's our solution? Our solution is corneal endothelial cell substitute from iPS cells called CLS1 cells. We can produce corneal endothelial cells from allogenic iPS cells with unified quality and stable supply. Also, we have developed cell transplant technique. Suspended CLS1 cells are injected into the cavity between cornea and lens called anterior chamber space. This surgery is very non-invasive, easy handling, and ready-to-use care. So here you can see the CLS1 proof of concept study with monkey bullous keratopathy model. The top left, you can see the corneal edematous thickness. The baseline is a normal corneal thickness. CLS1 transplanted eyes in blue significantly reduced corneal edematous thickness compared to the negative control eyes without cell transplant. At the same time, the cell transplanted eyes dramatically recovered transparency compared to the negative control eyes without cells. Also, we confirmed that the transplanted CLS1 cells cover the posterior surface of cornea. So, CLS1 transplant reduced corneal edema, recovered and normalized corneal thickness, and recovered corneal transparency dramatically. So what's our competitive advantages and IPs? First, we can produce CLS1 cells from allogenic iPS cells in just 1 month by our unique direct differentiation induction method. Secondly, we can produce and we can treat more than 1,000 patients per batch. And thirdly, we can preserve the cells as frozen stocks, which allows us to distribute the cells to medical institutions. These competitive advantages are totally protected by 6 patents. And here you can see the annual new patient forecast. The patient's number of bullous keratopathy forecast to steadily grow until 2030 to 1.2 million per year due to the growing country medical level, intraocular surgery increase, and population of aging. And here you can see the competitive landscape. We are the pioneer of iPS cell-based cell therapy for bullous keratopathy. Although there are some competitors who use cultured corneal endothelial cells from donor cornea, but the cell number from donor cornea is limited, and the cell quality varies due to the donor variation. By contrast, we can mass produce the CLS1 cells with stable quality, and we can preserve the cells, as I mentioned before. So what's the launch schedule? We will launch the cells in Japan in 2026, in US in 2029, and the rest of the world in 2030. Okay, so that's all. So if you are interested in our works, please contact us with URL code. Thank you very much for your kind attention.
Thank you very much for your presentation. I would now like to introduce the next speaker, Mr. Andy E., Director of Global Business Development from Human Life Co. Japan Inc. Andy-san, please start your presentation.
Hello, everyone. Thank you for coming to our presentation. My name is Andy Yi, Director of Global Business Development. Together, we also have Masamitsu Halata Shacho, CEO of Human Life for Japan. Currently, we face two major social issues in Japan. One, the number of incurable disease has reached to 338. Two, the nursing care cost by the government has reached to 12.3 trillion yen. Our vision is to contribute to the realization of an enriched society by playing a part in the development of umbilical cord tissue-derived cell therapy, therefore, to improve people's quality of life. Our mission is to achieve early commercialization of umbilical cord-derived cells step by step. Step one, by 2025 to achieve in acute inflammatory disease with serious illness, for example, long GVHD, ARDS, RID, and acute GVHD. Step two, by 2030 to achieve in chronic inflammatory disease with low quality of life, for example, CLD, sarcopenia, and chronic GVHD. How to achieve is to utilizing virtual pharma model together with strongest partner in each field. One, collaborative study with Institute of Medical Science, University of Tokyo, and Graduate School of Medicine of Nagoya University. HLC has the global rights to specific indications with UCMSC, IP, and manufacturing know-how, also patent application based on some preclinical work. Two, raw material procurement with IMSOCORT. Three, cell processing manufacturing with Roto and IMSOCORT together. Four, distribution by Alfresa. Five, clinical trial regulation submission with SRD. Six, commercialization either by our own cells. Actually, currently we're seeking partners for this field. In terms of global partnership, we're under confidential agreement with a leading US partner, which please stay tuned for a public PR announcement in the near future. We're here pioneering cellular medicine with UCMSC supply chain platform, starting with umbilical cord collection and stocking by IMSOCORT with strict requirements and high standards to ensure the best quality starting from the beginning. HLC together in conjunction with IMSOCORT established the Academic Industrial Hybrid Bank, also known as Public and Private Bank, with PMDA required cGMP manufacturing process, including QC tests, to ensure the best quality cell processing facility for master cell and working cell bank to scale up manufacturing. Fully integrated with IMSOCORT banking to receive umbilical cord tissue, partner with CMO for production and wholesaler for distribution in Japan. Even with a decreasing birth rate in Japan, our model will generate 1,500 doses from one input. also partnered with US banking company with local production for local consumption. International standardization for manufacturing process would take the world of cell therapy and regenerative medicine to ensure us to deliver the same high quality of product to the patients to achieve improved quality of life. Our direction of global UC-MSC production are executed in the following steps. Standardized manufacturing process, including QC tests between FDA and PMDA. For Japan, we have University of Tokyo MSET. For US, we have a leading partner with decades relationship to conduct US-Japan collaborative clinical study. After that, we expand into APAC countries based on local production for local consumption philosophy. Umbilical cords provides excellent usability and superiority, very low dependence on imports. Collection are done at baby's birth, domestic in Japan, where the cord normally are discarded materials, which also give us very low invasiveness and exposed risk to environmental factors compared to bone marrow and adipose. It has the highest migration capability inside the patient's body with special factors such as IDO PGE2 for inflammation and HGF BDNF for brain nerve recognition. Lowest immunogenicity in which avoid undesired response from one's immune system. Public-private bank available for both autologous and allogenic uses. The ability to freeze the core tissues for a long period of time will ensure the supply can satisfy the ongoing demand. We have achieved efficacy signal with no safety issue and excellent tolerability post UC-MSC infusion. Phase 1 for patients with steroid refractory acute GVHD have overall response rate of 83%, complete response with skin, liver, and gut 50%. Next slide. For our pipeline, we also have ongoing phase 1 COVID-19 ARDS, where we have received national grant from the Japanese government. We're ready for pivotal phase 2 in long GVHD. We have animal proof of concepts in multiple indications, such as secondary sarcopenia. For sarcopenia alone, there are more than 3 million patients. Next slide, please. Financially, we're healthy with $5 million cash, also in Series B. Why we're here? We're here to raise awareness, to have strong partners to help us to achieve our goals, because patient cannot wait. Thank you everyone again, and the event host, sponsors to give us this great opportunity to present. We welcome your interest and questions. Halata Shacho will be available to answer your questions in Japanese. Thank you very much.
Thank you very much to all the speakers. They were impressive presentations, really shining a light on emerging technologies that are coming out from Japan. Please feel free to contact the speakers for any inquiries. Thanks all. Have a good day. Bye.